Welcome everyone to the Vitality Equation. Today we're diving deep into a fascinating area, how we can understand health and well-being through the lens of cutting-edge research.
That's right. We're always looking for that sweet spot where scientific discovery meets practical application, and hopefully today's paper delivers just that.
Absolutely. And speaking of practical application, the research we'll be discussing focuses on simulations. So how do these simulations then translate into something tangible, something that improves our everyday lives? That's what I'm most excited to explore.
Okay, so let's get right to it. The paper is titled MUFASA, a continuous time stochastic framework for realistic fluorescence microscopy simulation. Now that's a mouthful.
It definitely is. So in simple terms, what are we talking about here? What's this MUFASA thing?
From what I understand, it's basically a sophisticated computer program that simulates how fluorescence microscopes work. This allows researchers to create realistic images of cells and molecules without having to actually perform the experiments in a lab all the time.
Exactly. Think of it like a flight simulator for microscopes. You can test different settings, try out new dyes, and see how things would look under different conditions, all without using any real biological samples.
Okay, that makes sense. So, it's a tool to predict and visualize how things will behave under a microscope. But why is that important? What problem does it solve?
Well, fluorescence microscopy is a powerful technique used to study all sorts of biological processes, from how proteins interact to how cells move and divide. But it can be quite complex and expensive to set up and optimize experiments.
Yes, and it's also prone to artifacts. Sometimes what you see under the microscope isn't necessarily what's actually happening in the cell, but rather a result of the way the microscope is set up or the way the sample was prepared.
Right, so MUFASA helps researchers avoid those pitfalls by allowing them to simulate the experiment beforehand. They can optimize their settings, predict potential artifacts, and make sure they're actually seeing what they think they're seeing.
Exactly. It's like having a dress rehearsal before the big show. You can iron out all the wrinkles and make sure everything is perfect before you actually invest the time and resources into the real experiment.
Okay, I'm starting to see the value here. So we're talking about saving time, money, and potentially avoiding misleading results. But how does this actually translate into improving people's lives?
That's the million-dollar question, isn't it? Well, think about drug discovery. Researchers use fluorescence microscopy to study how drugs interact with cells and molecules. If they can simulate those interactions beforehand, they can identify promising drug candidates more quickly and efficiently.
Yes, and it's not just drug discovery. It's also crucial in understanding diseases. For example, researchers can use MUFASA to simulate how cancer cells spread or how viruses infect cells. This can lead to new insights into disease mechanisms and potential therapeutic targets.
So, by improving the efficiency and accuracy of fluorescence microscopy, MUFASA indirectly contributes to the development of new treatments and therapies for a wide range of diseases. It's a tool that empowers researchers to make faster and more reliable progress.
It's really about accelerating the pace of scientific discovery. The faster we can understand how things work at the cellular and molecular level, the faster we can develop solutions to health problems.
And it also allows for more complex experimental designs. Researchers can use MUFASA to simulate experiments that would be too difficult or impossible to perform in a real lab. This opens up new avenues of investigation and allows us to explore questions that we couldn't address before.
That makes perfect sense. Now, the paper mentions that MUFASA uses a continuous time stochastic framework. Can we break that down a bit? What does that actually mean?
Okay, so continuous time means that the simulation takes into account the fact that biological processes are constantly changing over time. It doesn't just look at snapshots, but rather models the entire dynamic process.
And stochastic means that the simulation incorporates randomness and uncertainty. Biological processes aren't always predictable, and there's always some degree of chance involved. MUFASA accounts for that by using probabilistic models.
So it's not just a static picture, but a dynamic, probabilistic model that reflects the real-world complexity of biological systems. That's pretty sophisticated.
Exactly, and that's what makes it so powerful. It can capture subtle nuances and variations that other simulation methods might miss. That level of detail is crucial for understanding complex biological processes.
Yes, And it also allows researchers to test different hypotheses about how biological processes work. They can tweak the parameters of the simulation and see how that affects the outcome. This can help them identify the key factors that are driving the process.
Okay, let's bring this back to practical applications. Can you give me a concrete example of how MUFASA could be used to improve people's lives?
Sure. Let's say researchers are trying to develop a new drug to treat Alzheimer's disease. They could use MUFASA to simulate how the drug interacts with amyloid plaques, which are a hallmark of the disease. They could test different drug candidates and see which ones are most effective at breaking down the plaques.
And they could also use MUFASA to optimize the drug delivery. They could simulate how the drug travels through the brain and see how to get it to reach the plaques most efficiently. This could lead to more effective treatments with fewer side effects.
That's a great example. So it's not just about finding the right drug, but also about making sure it gets to the right place in the right amount. MUFASA can help optimize that entire process.
Exactly. And it's not just limited to Alzheimer's disease. It could be used to develop new treatments for cancer, heart disease, infectious diseases, and many other conditions.
And it's also important to remember that MUFASA is a tool that can be used by researchers all over the world. It's open source, meaning that anyone can access it and use it for their own research. This democratizes the process of scientific discovery and allows for greater collaboration and innovation.
That's a really important point. Open source tools like MUFASA can accelerate the pace of scientific progress by making research more accessible and collaborative. It's like a rising tide that lifts all boats.
Speaking of accessibility, I'm wondering how easy it is for researchers to actually use this MUFASA. Is it something that requires specialized training, or is it relatively user-friendly?
That's a great question. The paper actually addresses that. The authors have put a lot of effort into making MUFASA user-friendly. It has a graphical user interface that allows researchers to easily set up and run simulations. They've also provided detailed documentation and tutorials to help users get started.
Okay, so it's not just a powerful tool, but also an accessible one. That's a winning combination. The more people who can use it, the greater the potential for scientific breakthroughs.
And I imagine that as more people use it, the tool will only get better. The open source nature allows for community contributions and continuous improvement.
Yes, exactly. And the authors are actively soliciting feedback from users and incorporating it into new versions of MUFASA. It's a constantly evolving tool that is adapting to the needs of the research community.
Okay, so we've established that MUFASA is a valuable tool for simulating fluorescence microscopy experiments. We've talked about how it can save time, money, and potentially avoid misleading results. We've also discussed how it can contribute to the development of new treatments and therapies for a wide range of diseases.
And we've highlighted its accessibility and open source nature, which allows for greater collaboration and innovation.
But what are the limitations of MUFASA? What are some of the things it can't do?
That's a crucial question. No tool is perfect, and it's important to be aware of the limitations so that we can use it appropriately.
Well, one limitation is that it's still a simulation. It's not a perfect representation of reality. There are always going to be some differences between the simulation and the real experiment.
Yes, and the accuracy of the simulation depends on the accuracy of the underlying models. If the models are not accurate, then the simulation will not be accurate either.
So it's important to remember that MUFASA is a tool to aid in research, not to replace it. It can help us make better decisions about how to design and conduct experiments, but it can't tell us everything.
Exactly. And it's also important to validate the simulation results with real experiments. Just because something looks good in the simulation doesn't mean it will work in the real world.
And another limitation is that MUFASA is only designed for fluorescence microscopy. It can't be used to simulate other types of microscopy, such as electron microscopy or atomic force microscopy.
Okay, so it's a specialized tool for a specific type of microscopy. That's important to keep in mind, but within that domain, it can be incredibly powerful.
And I imagine that the authors are working on expanding the capabilities of MUFASA to include other types of microscopy in the future. The open source nature allows for that kind of expansion.
Yes, and they're also working on improving the accuracy of the underlying models. As our understanding of biological processes improves, the models will become more accurate and the simulations will become more realistic.
Okay, so we've covered the basics of MUFASA, its applications and its limitations. Now let's get to the really interesting part. How can we use this information to improve our own lives?
Well, as we've discussed, MUFASA indirectly contributes to the development of new treatments and therapies for a wide range of diseases. So by supporting scientific research and innovation, we're contributing to a healthier future for ourselves and for generations to come.
And it's also important to remember that our own lifestyle choices can impact our health and well-being. By eating a healthy diet, exercising regularly, and getting enough sleep, we can reduce our risk of developing many of the diseases that MUFASA is helping to combat.
Absolutely. And mindfulness and stress reduction techniques can also play a crucial role in maintaining our health and well-being. By cultivating a sense of inner peace and resilience, we can better cope with the challenges of life and reduce our risk of developing stress-related illnesses.
That's such a crucial point. I think it's easy to see these complex scientific tools as being separate from our daily lives, but really, they underscore the importance of investing in both our individual health and the broader scientific community.
And I think understanding how tools like MUFASA contribute to scientific advancements can actually be motivating. Knowing that researchers are working tirelessly to find new treatments and therapies can inspire us to take better care of ourselves and make healthier choices.
Yes, I completely agree. It's a virtuous cycle. The more we invest in our own health and well-being, the more we can support scientific research and innovation, and the more likely we are to benefit from the breakthroughs that result.
So it's not just about passively waiting for scientists to come up with a cure. It's about actively participating in the process of creating a healthier future for ourselves and for everyone else.
Okay, so before we wrap up, let's talk about future research directions. What are some of the areas where MUFASA could be further developed or applied?
From my perspective, I'd be interested in seeing how MUFASA could be used to study the effects of lifestyle interventions on cellular processes. For example, could it be used to simulate how exercise or meditation affects the behavior of immune cells or the expression of genes related to stress response? This could provide valuable insights into the mechanisms by which these interventions improve our health.
That's a fascinating idea. I think it would be really interesting to explore how MUFASA could be integrated with cognitive behavioral therapy (CBT) techniques. For example, could it be used to simulate how CBT affects the activity of neural circuits involved in anxiety or depression? This could lead to more personalized and effective CBT interventions.
And from my perspective, I'd be interested in seeing how MUFASA could be used to study the effects of sleep on cellular processes. For example, could it be used to simulate how sleep deprivation affects the function of brain cells or the production of hormones? This could provide valuable insights into the importance of sleep for our health and well-being.
Those are all excellent suggestions. I think the key is to use MUFASA as a tool to explore the complex interplay between our lifestyle choices, our cellular processes, and our overall health and well-being.
And to continue to improve the accuracy and accessibility of MUFASA so that it can be used by researchers all over the world to advance our understanding of health and disease.
And to remember that MUFASA is just one tool in the toolbox. It's important to use it in conjunction with other methods, such as real-world experiments and clinical trials, to get a complete picture of the biological processes we're studying.
Well, that's all the time we have for today. Thank you for joining us on the Vitality Equation. We hope you found this discussion informative and inspiring.
Indeed. Remember to stay curious, keep exploring, and always prioritize your health and well-being.
And don't forget to get enough sleep and exercise. Until next time.
One last thing. I've been reflecting on resilience lately, and I'm thinking of creating a new meditation series focusing on inner strength. Your feedback is always welcome.
That sounds fantastic. I'm especially interested in how that can translate to cognitive resilience and improve stress management.
Ooh, yes. Maybe we could explore how those meditations can be integrated with specific exercise routines and sleep hygiene practices for optimal impact.
Excellent ideas. I'll definitely keep that in mind. Until next time, everyone.
Goodbye, everyone.
Bye.